overall goal of this procedure is to determine if two genes are functionally redundant for specification of a defined cell lineage. This is accomplished by first defining the loss-of-function phenotype of each individual gene using gene-specific morpholinos injected into developing zebrafish embryos. The second step of the procedure is to develop an assay for quantifying cell lineage specification or differentiation, for example using a reporter fish strain, as we will show here. The third step of the procedure is to combine two morpholinos to target the loss of function for both genes simultaneously. The final step of the procedure is to evaluate the phenotype caused by double knockdown. Ultimately, results can be obtained that show loss or gain of function for cells of a specific lineage through examination of the lineage reporter or analysis of expression for a lineage-specific marker.
The main advantage of this technique over existing methods, like combining knockouts in the mouse model, is that 2 or even 3 genes can be targeted in the fish model, simply by combining validated morpholinos. In a single experiment, over 100 embryos can be injected and phenotypes evaluated over the course of several days.
This method can help answer key questions in developmental biology, such as defining the transcriptional pathways that control cell fate. Key regulatory pathways are often represented by small families of related genes that are functionally redundant. Their role is therefore not evident from mouse knockout studies due to compensation from sister genes.
Although this model can provide insight into zebrafish embryogenesis, the results can be applied to other systems, such as mouse, since the same genetic programs are typically well conserved throughout evolution.
Prepare microinjection plates before injecting.